Hello everyone, so today in this video we are going to do the mechanism of cell injury. In cell injuries we are going to do three things initially, and after that we're going to explain each and everything. So in cell injuries, first of all we will check the cellular response to that kind of to the injury. For example, we will check the duration, the severity, and the type of cell injury. After that we're gonna check the response of that particular cell to the injury by checking at the status, adaptability, and genetic makeup of that particular cell. For example, the skeletal muscle can survive like 2 to 3 hours without the oxygen or with the ischemia, while the heart muscles can only survive 20 to 30 minutes, and as well as the brain cells can only survive like for a few minutes. Okay, and after that we're gonna also check like the physical and biochemical changes. Because of that there's gonna be cell injury. So the physical and biochemical changes are reduced level of the ATP, increased level of the reactive oxygen species. There's going to be increased intracellular calcium level. There's going to be membrane damage, as well as there's going to be DNA damage and increased level of the misfolded protein. Okay, now we're going to do first the reduced level of the ATP. In reduced level of the ATP, what's going to happen? For example, there is ischemia. So because of the ischemia, the oxygen level is reduced, as we know that. So there's going to be reduced level of the oxidative phosphorylation. Because of that there's going to be reduced ATP. So when the level of the ATP is less, so because of this there's going to be mainly two things, not two things, like there's going to be mainly three things. First, there's going to be the sodium potassium ATPase will not work, and because of this there's going to be increased level of these calcium and sodium and potassium inside the cell. And because of that, we already studied in second year that there's gonna be thing called solvent drag, and because of this the water is gonna be coming inside the cell, and because of this the cell gonna swell. Apart from this, there's gonna be anaerobic glycolysis. Because of this there's gonna be reduced level of the glycogen. Lactic acid level will be increased, as well as there's gonna be a reduced level of the pH, because it's going toward the acidity. And ultimately, because of the reduced level of the ATP, because the reduced level of the pH, there's going to be clumping of the nuclear chromatin. Okay, now the third thing is that there's going to be detachment of the ribosomes from the rough endoplasmic reticulum. Because of this there's going to be reduced level of the protein synthesis, and ultimately there's going to be cell injury. Okay, now we already the second point there was the increased level of the reactive oxygen species. So we will first we all I missed one point, that there's going to be mitochondrial damage as well, but you can put it over here, the second point. What happened in mitochondrial damage? For example, we already said that because of the ischemia there's going to be reduced level of the oxidative phosphorylation. Because of that there's going to be reduced level of the ATP and increased level of the reactive oxygen species. Because of that there's going to be increased abnormality and ultimately necrosis. And as well as we already studied that mitochondria is also related with the apoptosis. So for example, there's going to be damage, so because of that there's going to be increased level of the pro-apoptotic protein. And because of this there's going to be reduced level of the anti-apoptotic protein, which are BCL2 and BSCLX. And because of this thing, the mitochondrial mitochondrial permeability transition pores will open, and because of this the cytochrome oxidase will come out of the mitochondria, and the cytochrome oxidase will be combining with the ApoF1, and the apoptosome will form. And further there's going to be steps of the apoptosis, and ultimately there's going to be apoptosis because of the mitochondrial damage. So in mitochondrial damage we did two things, the necrosis and apoptosis. Now we are going to do the increased level of the calcium in the cell. So the calcium will be coming from outside, like the influx of the calcium will be really high, as well as the calcium will come from the mitochondria, as well as from the endoplasmic reticulum, mainly the smooth endoplasmic reticulum. Because of this the cytoplasmic calcium level will be increased, and ultimately there's gonna be two things. First, there's gonna be we already said it, that there's gonna be transition pores. So increase permeability transition, as well as increased level of the different kind of enzyme. Like there's going to be increased level of the phospholipase, endonuclease, protease, and ATPase. And because of phospholipase, phospholipid level will be reduced. Endonuclease cause destruction of the nucleus, and protease will cause destruction of the protein, and ATPase will reduce the level of the ATP. And ultimately, all of these things are going to cause the injury, membrane damage. Okay. Now we're gonna do the we already we there was a second point in the list. We already did it, that there was an increased level of the reactive oxygen species. So for example, if there is normal oxygen, so in injury there are normal oxygen in normal cell, the normal oxygen is going to be converted into the superoxide. This is superoxide. The superoxide, in the presence of superoxide dismutase, it's going to be converted into hydrogen peroxide, H2O2, and in the presence of um, it's in the presence of nitric oxide, it's going to be converted into pyronitrate. Then the superoxide H2O2 is going to be converted into OH in the presence of Fenton reaction. And if we talk about the leukocytes and the neutrophil cells, what's going to happen, this the oxygen is going to be converted into superoxide, and superoxide is going to be converted into H2O2 in the presence of superoxide dismutase. And after that, there's going to be enzyme, this is called myeloperoxide peroxide dismutase. It's going to convert the in the presence of Cl chloride, HOCl. Okay. Now there are two terms which we have to study. First is respiratory burst, and third is oxidative stress. Respiratory burst is when there is excessive release of reactive oxygen species, and oxidative stress when there is a very high level of the reactive oxygen species. So what are the causes of reactive oxygen species? There's gonna be radiation, chemical, as well as the inflammation. Now we're gonna do the, like, we it can come in the paper as well, like what are the fate of the reactive oxygen species. So the superoxide is going to be either converted into H2O2, which is less toxic in the presence of superoxide dismutase. Either they're going to be converted into H2O in the presence of glutathione peroxidase. Either they're going to be converted into H2O plus O2 in the presence of catalase, or in the presence of antioxidant ACE and beta carotene. They're also going to be destroy it or reduce its level. Okay. Now if we talk about the mechanism, what actually the reactive oxygen species do, they're gonna do three things. They're gonna cause membrane damage by lipid peroxidase. Say by lipid peroxidase, there's going to be membrane damage, and there's going to be protein modification. Because of that, there's going to be damaged protein, as well as there's going to be DNA mutation, and because of that there's going to be DNA damage. Now we are going to do the last thing which we did, that like the damage to the membrane permeability. It's because the membrane damage, so it's because increased level of the phospholipase, reduced level of the phospholipids, as well as there's going to be increased level of the lipid breakdown, and reactive oxygen species are really high as well. And because of that, there's going to be membrane damage. We did four things. What were they? Increased level of the phospholipase, reduced level of the phospholipid, increased level of the reactive oxygen species, and increased level of the lipid breakdown. Okay, now we're going to do the last thing in this topic, like what is autophagy. So for example, there's going to be nutrient deprivation in the cell. So because of this, there's going to be autophagic vacuole will form. Autophagic vacuole is going to be combining with the lysosome, and ultimately they are going to be forming autophagolysosome. And in the end, the autophagolysosome will cause destruction lysosome will release different kind of enzymes and reactive oxygen species, and there's going to be destruction. This is called autophagy. Thank you.